clickers and get ready for exceptions to Mendel and Pedigrees. We'll start with a clicker question. Two deaf parents with homozygous mutations have a child with normal hearing. Which of these answers could explain this? Read through the answers and give it your best guess. Okay, if you're not sure, just go ahead and pick an answer so we can move through this. Let's see what you guys think. A diversity of opinions. So A and E were the least popular, B, C, and D people liked. So A, the gene causing deafness is dominant. That's not really going to work because both parents are homozygous. So they're both going to give the dominant allele to the kid and the kid is not deaf. So we can't use B. E, the gene involved exhibits codominance. That doesn't really make sense. There's only just talking about being deaf. It's not a good answer. So we got to choose between B, C, and D. The parents have two different alleles of the deafness gene. Maybe, we'll talk a little bit about in lecture why that might be true, but that's not my favorite answer. C, the child is heterozygous for two different genes that cause deafness. That's my favorite answer. I think this is the best answer. The other ones could be right. That's what we're going to talk about today, is epistasis. D, the gene involved exhibits incomplete dominance. Not so good. Incomplete dominance is when the heterozygote has an intermediate phenotype. But both the parents are homozygous, so the child isn't going to be a heterozygote. So D is not very good. You can make an argument for B, but C was the answer that I think is best. It's not an exam question, it's a clicker question. So why is C possible? Because there's multiple steps in hearing. People who are deaf can have defects at several different levels. They could have conductive hearing loss, and that's where there's problems transmitting the vibrations to the inner ear. So they could have a problem with the bones of the inner ear, for example. There's sensory neural hearing loss. That's getting that signal in the inner ear through the nerve. So they could have a problem there. There's also central hearing loss, where there's a problem in the area of the brain that perceives the sound. So one explanation would be one parent had a conductive hearing deficit, and the other parent had a sensory neural hearing deficit, and the child that was born was heterozygous at both of the loci controlling those traits and ended up hearing just fine. So that's what we're going to talk about today is the principle of epistasis. And I didn't give you this slide because I'm not expecting you to memorize or understand beyond what I just told you of hearing loss. So epistasis, I think, is a common sense principle, but because we call it something fancy like epistasis, people tend to get confused about it. So I'm going to define the term a couple different ways and work through a couple of examples, and hopefully by the end of all that it will make sense. When the phenotype, the principle of epistasis is that the phenotype associated with alleles present at one locus can mask or alter the phenotype controlled by alleles at another locus. So there's two different genetic loci controlling the same trait, and that causes this epistasis. And this is true in many, many, many cases because most traits are controlled by more than one gene. Remember, that's why I made up the favorite ice cream flavor locus, because I couldn't find a human trait that was truly controlled by one gene. So there are some, but I made one up just to make it easy. So many traits are controlled by more than one gene. The results of epistasis are listed on this slide. When two genes affect different steps in the same pathway, when you do a dihybrid cross, the phenotypic ratio is not 9:3:3:1 anymore. That's a hallmark of epistasis. You get a modified 9:3:3:1 ratio. And when we go through the examples, I think it will become clearer what that means. A key point is that the genotypic ratios in the dihybrid cross don't change at all. When you do your Punnett square, it turns out exactly the same, whether there's epistasis or not. The trick is how you assign the phenotypes to the different genotypes. So this is an alternate definition, a little bit more sciencey. Epistasis occurs when there is phenotypic interaction of non-allelic genes. One gene can mask another. So the genes are non-allelic because they're not two alleles of the same gene. They're different genes, so they are non-allelics. They're non-allelic genes. They're not two variants of the same gene. They are different genes altogether when we're talking about epistasis. Okay, example number 1 is epistasis in flower color in peas again. So we have two true breeding parents. They're both white. This one is homozygous dominant at the C locus and homozygous recessive at P. This other true breeding white plant is homozygous recessive at C, homozygous dominant at P. When you cross these to get the F1 generation, they are all dihybrids. They're heterozygous at both loci. And they're all purple. Because as long as you have one functional allele at each locus, you can get all the way to purple. Now I provided you with a molecular explanation of this phenotype because maybe that will help it make sense. I said, okay, maybe there's an enzyme C that converts this white substrate to another white substrate. That's what enzyme P works on, and enzyme P turns this white substrate purple. So if you're missing enzyme C, enzyme P has nothing to turn purple. And if you're missing enzyme P, this intermediate has no color. So either way, if you're missing C or P, you end up white. Now when you cross these F1s to make the F2 generation, you do a dihybrid cross, standard Punnett square, just like Friday. All these gametes are going to show up with equal frequency, so you get the genotypes in the Punnett square. But now we have to assign the phenotypes. And remember, if you don't have C or you don't have P, then you end up white. So what we see are 9 pea plants out of 16 that got at least one dominant allele at both locus, and 7 that got at least 2 recessive alleles at one locus. After we go through the next couple problems, I think why you assign the phenotypes this way will make a little bit more sense. And I'll stop for questions then, if you're confused. So this is example number 2. It's coat color in Labradors, another classic example of epistasis. So there's 2 genes involved here. The first one is E. The E locus controls eumelanin production. Eumelanin is the pigment that's going to turn the hair black or brown. So if you have a dominant allele, you make eumelanin, and it can turn the hair into a pigmented hair, either black or brown. If you have a little e, you don't make any melanin, and you end up being a golden color, or a yellow lab. At the B locus, there's an allele that controls melanin disposition in the hair. So if you have at least one dominant allele, you can deposit lots of melanin in the hair. If you have the recessive, less functional allele, you can't put as much melanin into the hair, so you end up being brown. So my question for you guys to practice here is, what are the possible genotypes of these dogs? Because what's tricky about epistasis is assigning the phenotypes. So go ahead and give it a try, and we'll go through the answer. If you're having troubles, I'm going to start with the yellow labs here. So do them first. Okay, anybody want to volunteer an answer for the yellow labs? What's the genotype? Yes, sir. Very good. What about the other locus? Excellent. So what he said, for those of you who could not hear, was these guys have to have two alleles. That's why they're golden colored, because they can't make any U melanin. And it does not matter what's at the B locus, because they don't have any U melanin to deposit in the hairs anyway. So once you've got two little E's, you know you're set. Now we've got to do the brown and the black. What do you know about the brown labs? They have to have what? They have to have at least one big E and two little B's. I heard that out there. So if you're going to be brown, you have to be homozygous recessive at the B locus. And of course, the black labs have to have at least one dominant allele at each of these loci. They have to make the pigment, and they have to be able to put a lot of it into their hairs to turn them black. So if you understand this and you're happy with it, fine. But something that really helps me is to use an abbreviation for these kinds of dihybrid crosses. So this double dominant phenotype has to have at least one dominant allele of each, and the second one, denoted by a dash, could be either a dominant or a recessive. It doesn't matter. They just have to have at least one. So all four of these phenotypes match with that abbreviation. For the browns, they have to have at least one big E and two little b's. And these guys would be either the homozygous recessive E with whatever one big B and whatever the dash is, or doubly homozygous recessive. And why labeling them this way helps me, I hope will become clear on the next slide, that this is an alternate strategy. So once you know this, you can deal with your dihybrid cross that you did using a Punnett square. The genotypes in here are exactly the same. It's describing the phenotypes of the genotypes that's tricky. So we get a modified ratio here. We have 9, 3, 3 plus 1. So that's how it's modified. You add up some combination of the 9s and the 3s and the 1s to get the new phenotypic ratio. And the reason why I like to do this is because that helps you figure out which numbers to add. So in any dihybrid cross, you're going to have 9 that have at least one dominant allele at each locus. You're going to have 3 that are dominant at one, recessive at the other, 3 that are recessive at that and dominant at the other, and 1 that's homozygous recessive at both. So now it's assigning the phenotypes to those genotypes. In this case, we know that if you have 2 little E's, you're going to be a golden lab. So we add the 3 and the 1. And that's how I got through this epistasis experiment. In the one we did before with the purple and white flowers, in that case, if you were recessive at either locus, this one, or both, you were white because you couldn't progress all the way to P. So in the first example, it's 9 to 7, and 7 is 3 plus 3 plus 1. That's how it's a modified 9, 3, 3, 1 ratio. Do we have any questions? Good. Okay. So this is a slide I showed you before, and last time I showed it to you, I said these plants are genetically exactly the same, but they were grown in different environments. So they have different phenotypes. It could also be that the environments are the same, and the genotype at one locus controlling flower color is the same. But there's another locus that affects flower color, and that's different in these two plants. So an important point that you need to take away about genetics is that genes at other loci and the environment affect the phenotype. Most traits depend on multiple genes, and the environment often affects how those genes are converted into a phenotype. Now last time I gave you a problem where I had 2 carriers of cystic fibrosis mutations, and they had different mutations. One of them was delta 508, and one was G542X, which has a stop, the premature stop. And I said, doesn't matter, if you have one of each of these alleles, you're still homozygous recessive and you have cystic fibrosis. Now I chose those alleles because both of these are very bad alleles. They produce almost no channel in the membrane. So people who have these alleles are very sick. But that's not true in all allele that you have can also affect phenotype. And I found this video that I like. It goes through a couple different ways you can get CF, and it calls to mind some of the things you've learned earlier in the class.
Many mutations of the CFTR gene can disrupt ion transport through CFTR channels. In general, cystic fibrosis-causing mutations reduce CFTR ion transport by affecting the quantity of CFTR channels at the epithelial cell surface, or by affecting the function of cell surface CFTR as an ion channel. Some CFTR mutations that reduce the quantity of CFTR at the cell surface lead to little to no CFTR channels or some channels reaching the cell surface. The degree to which the channels are absent often relates to the severity of disease. Mutations that cause little to no CFTR at the cell surface are often the result of improper folding and processing of the CFTR protein. The most common CF-causing mutation, F508 deletion, is an example of this type of mutation. Premature stop codon mutations, such as G542X, also generally result in little to no CFTR at the cell surface. Without functional CFTR channels at the cell surface, there is very limited or no chloride ion transport, leading to electrolyte and fluid imbalances throughout the body. Patients with 2 of these mutations often have more severe CF disease, including pancreatic insufficiency. Other CFTR mutations may reduce the quantity of channels at the cell surface, but to a lesser degree. Often, these mutations are the result of errors in RNA splicing, leading to a reduced but variable quantity of functional CFTR at the cell surface. A common example is the 3849 plus 10 kb C to T mutation. Generally, such mutations are associated with some residual ion transportability. However, it is not sufficient to maintain proper homeostasis. Those patients who carry at least one such mutation may have a more moderate CF phenotype and are more likely to be pancreatic sufficient. Other CFTR mutations lead to a normal quantity of CFTR channels at the cell surface. However, their function is reduced. These mutations can affect the ability of CFTR channels to open, known as a gating defect, or the ability of ions to flow through the channel, known as a conductance defect. Gating mutations often reduce the function of CFTR channels to a greater degree. Such channels rarely open to allow ion passage. The most common example of this type of mutation is G551D. These mutations are often associated with severe CF disease and pancreatic insufficiency. Conductance mutations reduce the rate of chloride flow through the CFTR channel. This is thought to be caused by a defect in the pore of the channel that restricts ion transport. The most frequent examples in this group include the R117H mutation. Patients who carry at least one conductance mutation often have a more moderate CF phenotype and may be pancreatic sufficient.
Okay, so that shows you how lots of the things we talked about in the molecular biology section can actually cause disease. If you're interested, there are a couple more videos about cystic fibrosis here that are nice. Okay, so to summarize what we just talked about, mutations in the same gene. How can you get different phenotypes? I don't think I gave you this slide. So the first one is you could have the same alleles, but differences in the environment. So that was the 2 hydrangeas. One's grown in acidic soil, one's grown in alkaline soil, and they get different colored flowers. Environment affects the expression of phenotype. You could also have the same alleles at one locus, but there's an epistatic interaction with genes at other loci. One other locus or multiple other loci could also affect the phenotype. So that can mask or alter the expression of the phenotype associated with these 2 alleles. Different alleles with different levels of function. So that's what we just went through with the CFTR mutations. Depending on what kind of allele you have, how much activity it has, it can alter the severity of the phenotype or other aspects of the phenotype, how the phenotype appears. So you can have same alleles and differences in environment and genes at other loci, or you can have different alleles to give you a different phenotype. Are there any questions? Because we're going to move on to something else. Okay. So now we're going to talk a little bit about pedigree analysis. This can help you figure out how different diseases are inherited. Usually what you're going to see is a phenotype pedigree when somebody comes into the doctor's office and they interview them, take a history, who in your family was affected with this disease, and they draw a pedigree like this. There's always a key or an explanation, and in this case the filled-in individuals have cystic fibrosis in this pedigree. Over here, what you're seeing is a genotype pedigree because it has these half filled-in shapes. Those aren't people who are halfway sick, those are people who are carriers. So these individuals are not affected with the disease at all. Instead, they're carriers. Sometimes you can infer this by looking at talking to people, looking at the pedigree, you can figure out who the carrier is, but sometimes you need a genetic test. So, for example, we know that one of these two grandparents, at least one, has to be a carrier. But we don't know which one unless we do a genetic test. So this implies a genetic test was done and we know it's grandpa, or from other pedigree information we know it's him. Some common symbols you're going to find in a pedigree. Filled in is pretty much always affected, whereas the open symbols are normal. Circles are for female, the squares are for men. Mating is a horizontal line, inbreeding is two horizontal lines, and offspring are little drop-down lines. So if there's multiple offspring drop down from the same line, it means that they're siblings. Now this is an example of a recessive pedigree. We know that you're seeing genotype because we've got these half-filled-in guys who are carriers. Some of the characteristics of a recessive pedigree, you often see that both sexes are affected with similar frequency. Most people who are affected have parents that are not affected. So here, remember, these guys are carriers. They're normal. So these two affected people have normal, unaffected parents. Same down here. Parents are not affected. Approximately 1/4 of the offspring of heterozygous parents are affected. So here, these people actually had 4 kids, and they had 2 that were affected. That doesn't mean it's not recessive inheritance. It just means they were unlucky. Instead of having 1/4 of their kids have the disease, they were unlucky and half of them did. It's just a random event like flipping a coin. And the parents of affected people are often relatives, so you'll see that there's this indication of inbreeding with, I think it's a cousin in this pedigree. And that is important because often these recessive disease-causing alleles are rare in the general population. So if you just marry some person off the shelf, they're probably not going to have this mutation, but if it's somebody in your family, the frequency of that recessive allele is higher in your family, so it's more likely 2 recessive mutant alleles will come together when there's inbreeding. Now there's a mistake in this pedigree. I have a reward for the first person to find it. Anybody see that? Yes, sir. Which generation is it? 1, 2, 3, 4. What do you see? Okay, he's right. The lower left corner there should be a heterozygote, right? Because if the number 2 there is affected, they have to have 2 disease-causing alleles, and they should have given them to their kids. So my husband is a pediatric geneticist, and sometimes this happens. It's a little bit embarrassing, but clearly there was somebody else on the scene when these 2 guys were conceived, because it couldn't have been him. Okay, everybody get that? Okay, on we go. Dominant alleles. So here we have a pedigree where there's no half filled-in people. All the filled-in guys are affected. Again, we see that both sexes are affected with equal frequency. In this case, every person has an affected parent. And that's because it only takes 1 allele to be affected. So if you pass it on to the next generation, you were affected too. Approximately half the offspring of an affected person are affected. Now remember we said this is a Huntington's disease pedigree. Why doesn't everybody have Huntington's disease if it's dominant? Because the allele is relatively rare. So because of that, we know that an affected person probably is heterozygous. Sometimes with a dominant, nasty mutation, 2 copies can be lethal too. But in this case, it's just that it's a rare, I think it's just a rare allele. So the heterozygotes are, the affected people are usually heterozygotes. Okay, questions, or we're good? So we're just talking about allele frequency, and I told you CF was more common in Caucasian populations than it is in African or Asian populations. And in each population of people in different races, you're going to have different diseases and syndromes and alleles that are more common. So I wanted to talk about another one. This is alcohol flush syndrome that's more common in Asian populations. So when this gentleman drinks alcohol, he looks like this. And I'm sure all of you know somebody who this happens to. Maybe it's even yourself. So I'm going to tell you why this happens. So when you detoxify alcohol, there's 2 enzymes you need. Alcohol dehydrogenase takes alcohol to acetaldehyde. Then acetaldehyde dehydrogenase 2 converts acetaldehyde to acetic acid, and then it's not so harmful. Acetaldehyde is a lot like formaldehyde, the thing that you pickles, organisms in jars, nasty stuff, carcinogenic, and it tends to cause esophageal cancer. So what does this have to do with genetics? Well, the ALDH22 allele predisposes you to esophageal cancer. The ALDH21 allele is a functional protein. The ALDH22 allele is an inactive protein. And this number 2 allele, if 40 to 80% of East Asians have at least one of these alleles. So it's very common. ALDH2 deficient individuals have a higher risk of esophageal cancer from drinking alcohol. This is an example of incomplete dominance as well, because if you're a heterozygote, your risk is halfway between a person that has the 1 allele and a person that has 2 copies of the 2 alleles. So heterozygotes have an intermediate phenotype. And what that means is if you flush, you're flushing because you have lots of acetaldehyde in your circulation, because that causes increased heart rate, flushing, and nausea. And that means that you're going to have more of this nasty formaldehyde-like thing in your stomach getting into your esophagus. So if you flush at all, you're predisposed to esophageal cancer. If you want to read more about this, these are 2 reputable sources that are links if you want to learn more. So why are bad, nasty alleles like this so common? That doesn't make much sense, right? What happened in natural selection? Well, it turns out that heterozygosity for CFTR mutations may have been helpful back when there was typhoid around. And this ALDH22 allele may help you be resistant to parasitic infections, the things that grow in the water, live in the water that rice grows in, because the allele maps with rice cultivation. Whereas the sickle cell anemia allele that we talked about, if you're heterozygous for that, it protects you from malaria. That's known. And if you look at the distribution of the sickle cell allele and malaria in Africa, you can see there's really good overlap, consistent with the idea that under certain conditions, these alleles are actually beneficial. So I have another clicker question for you here. Pediatrician sees 2 patients with CF in the same afternoon, but one is much sicker. Which of the following could explain this? The patients carry different alleles. One patient is heterozygous, epistatic interactions, or one patient is homeschooled. Go ahead and give it a guess. Okay, click in if you have it. All right, so a lot of you guys like C. I'm surprised, because I just told you all about this. The patients carry different alleles, right? But actually, any of these answers are right except B. And you guys laughed at this, but why is homeschooled relevant? Yeah, they're not getting infections because they're at home. They're not exposed to many kids, so they get less scarring in their lungs. So nice job. Enjoy your break.